Hello, this is AJ Williams, and today I will be talking about olfactory dysfunction, specifically how olfactory dysfunction has been found to be an indicator of and a potential cause for certain neurodegenerative diseases such as Parkinson's and Alzheimer's. First, I want to start off by talking about how the neural processes of the olfactory system work. Messages are sent to the bilateral olfactory bulb, which lies at the base of the orbitofrontal cortex. It has been described as a sort of olfactory thalamus, given that it is the last stop for sensory processing before being sent to the cortex. Within the olfactory bulb are several layers of neurons, the first being the olfactory epithelium. Lying at the top of the nasal cavity, these neurons are responsible for picking up chemical signals in the nose to later be perceived as scents. The axons in these olfactory receptors are surrounded by olfactory ensheathing cells, or OECs. Much like Schwann cells, macrophages and microglia are found in populations between these OECs. Their purpose is to shy away harmful chemicals, viruses, and xenobiotics. The olfactory receptor's axons synapse in the glomerular layer, which holds a synapse that is shared by several different axons, usually 3 or 4 different types. The main neurotransmitter, transmitted from the olfactory receptor cells to the next layer, the mitral cell layer, is glutamate. The synapse between the olfactory receptor cells and mitral cells is also shared with juxtaglomerular cells and centrifugal fibers. Both of these act to inhibit and therefore mediate the activity of both the presynaptic olfactory cell and the postsynaptic mitral cell. The glutamate that is released from the olfactory cell can bind to NMDA and AMPA receptors on both the juxtaglomerular cells and the mitral cells. When bound to the juxtaglomerular cells, a mechanism is put into motion to release GABA into the synapse. These neurotransmitters then bind to the presynaptic GABA receptors, which then reduce the amount of calcium intake, which in turn will reduce the amount of glutamate released from the presynaptic cell. The centrifugal fibers release serotonin, which binds to receptors on the postsynaptic mitral cell and indirectly hyperpolarizes it, thereby inhibiting its firing. From the glomeruli, mitral cells send their signals to the anterior olfactory nucleus, or the AON, from which many of the centrifugal fibers in the glomeruli originate. The signals are then transmitted to the olfactory cortex. Notice that throughout the explanation of the neural pathway, I never mentioned the blood-brain barrier. This is because the path from the receptor cells in your nasal cavity to the olfactory cortex enables viruses and xenobiotics to directly enter your central nervous system, effectively bypassing the blood-brain barrier. As mentioned before, microglial cells in the nasal cavity work to prevent these harmful stimuli from entering the brain from the nose. Now let's move on for a moment to describe why these microglial cells are important. An area of the brain known as the locus coeruleus is the source of noradrenergic projections that synapse in both the olfactory bulb and in the forebrain region associated with smell. Norepinephrine is readily transmitted through noradrenergic pathways in microglia, and it is primarily responsible for regulating the permeability of the blood-brain barrier. There is also evidence norepinephrine aids in learning new odors. A deficiency in norepinephrine may be in part a cause in hyposmia, a deficiency of smell, because the microglia in the blood-brain barrier may be inhibited in their ability to stop harmful substances from infiltrating. In patients with Parkinson's disease, the locus coeruleus is significantly impaired, largely by deposits of great amounts of alpha-synuclein and Lewy bodies. This may be a factor in the 90% of Parkinson's cases that exhibit some form of hyposmia or anosmia. Anosmia is very rare in these cases. I will get back to alpha-synuclein in a moment, but first let's look at a few other reasons that have been explored for the olfactory dysfunction in neurodegenerative diseases, namely Parkinson's. The size of the olfactory bulb was measured in some experiments in both people with and without Parkinson's, both in the same age range, of course. Overall, people with Parkinson's had smaller olfactory bulbs, and there was significant evidence that a smaller olfactory bulb correlated with decreased overall olfactory performance. However, the relative performance of the 3 tests of olfaction, being odor discrimination, odor threshold, and odor identification, showed varying and inconclusive results. Overall, it is not clear whether Parkinson's was the direct cause of the reduced size of the olfactory bulb, or if it was just age, stress, environmental factors, smoking, or maybe acute major depression. Schizophrenic patients were tested against control groups in several different tests with the same parameters: odor acuity, odor threshold, and odor identification. In these tests, the schizophrenic patients scored, on average, half a standard deviation below the mean for odor acuity and a full standard deviation below the mean for odor identification. Schizophrenic patients performed worse in all studies than the controls. This may give strong evidence for olfactory dysfunction in a schizophrenic person, but the results of these tests were, with the first order relatives, failed to show any evidence that olfactory dysfunction can be an indicator to diagnose schizophrenia, much like it can be used as an indicator for Parkinson's. Now let's revisit the deposits of alpha-synuclein found in the locus coeruleus. For now, I'll refer to it as alpha-syn. First, I should clarify that alpha-syn deposits are found in many areas of the brain in Parkinson's patients, but are especially pronounced in the locus coeruleus. Alpha-syn is a protein that, when misfolded, can develop into Lewy bodies, which are large proteins that waste space in the cytoplasm of cells. Parkinson's, along with Alzheimer's, is considered a Lewy body disease because Lewy bodies are so heavily dispersed throughout the brain and inhibit function. Alpha-syn pathology is thought to be the first symptom of Parkinson's, starting in the olfactory bulb and spreading throughout the rest of the brain. This would explain why we tend to see olfactory dysfunction early on as one of the first symptoms. Another theory for olfactory dysfunction is tau-related pathology. Tau is generally a protein that regulates microtubule assembly, but can lead to the formation of fibrillary tangles, which are common in both Alzheimer's and Parkinson's disease. Tau-related pathology tends to be seen in many of the same areas as alpha-syn in patients, indicating they may be functionally related. But more importantly, it occurs in the bulb of the anterior olfactory nucleus in Parkinson's, Alzheimer's, and Lewy body dementia patients. All of these diseases traditionally have an olfactory dysfunction component, as opposed to progressive supranuclear palsy and corticobasal degeneration. These patients usually have minimal or no olfactory loss, and tau-related pathology does not seem to occur in the anterior olfactory nucleus in these patients. These data indicate that tau could be a central contributor for olfactory loss or dysfunction. Lastly, I want to mention that these are all theories. Though some correlations have been found, we do not know for sure what precisely causes olfactory dysfunction in neurodegenerative diseases, because most of the time when a person is diagnosed, they had already lost some of their sense of smell long before their diagnosis. It is possible that the cause for some of these olfactory deficits is just environmental factors. Many of the risk factors for Parkinson's and some of the risk factors for other neural diseases are the same risk factors for loss of smell that is unrelated to neurodegenerative disease, namely age, head trauma, exposure to nanoparticles, viruses, ionized metals like manganese, and pesticides. This points to the possibility that damage to the olfactory tract and bulbs may play a role in leading to the cause of some of these neurodegenerative diseases. But at the end of the day, if we don't know the direct cause of olfactory dysfunction in neurodegenerative diseases, the knowledge that it is widespread in many neurological disorders is useful. This can help to diagnose people as early, as symptoms like these are exhibited sometimes up to a decade in advance in Parkinson's patients especially. If possible, it would be wise to incorporate some sort of smell check into checkups, just as the reflexes in your eyes are checked. Seeing as olfactory dysfunction is one of the first symptoms of many neurological diseases, it seems wise that this would be the next step to take to ensure people are diagnosed early.